Hi everyone, welcome back to my channel Bio Beavers, and here's Dr. Pragya helping you with all the bio solutions. I know I am uploading video after a very long time, but I promise there won't be any more delays and you'll see more and more videos which will help you out with a lot of doubts. Without any further more discussion, I'm going to start today with paper 5. And I know I've already uploaded a few paper 5s on my channel and you can see the videos. So we'll start today with the Feb March version. So we'll start today with the Feb March 2024 version. And we'll start with paper 5, which is the planning and analysis evaluation paper. Before I move on to the paper, there are a few pointers which I've always pointed out in my previous videos. Also, it's a 30-mark paper. It will have two questions to it. Each of the question has certain parts which are highly scoring, and there are certain tips which I have shared in the earlier videos on my channel a few months ago, and we'll start with this paper. So, the first question, as I mentioned, the first question usually starts with one page, as you can see, of certain amount of data which has been given to you. And there will be a lot of information which will be given to you in this process. And while we are working on that, please remember to underline the important points. Okay, so there are important points which should be underlined in this, so that when you go back to the question, you can just come back and you can refer to each one of them while you are actually attempting the questions, and it doesn't take a lot of time with that process. So in this paper, if you have a look at it, it's about the onion. It says onions are vegetable that are harvested from the plant Allium cepa. In many countries, small onions are used to make pickled onions. So, as I mentioned, this part should be like a fast read, like you're reading the novel. Just try and read it once, which is like a fast read, and underline the things, which you think might be important, and we might come in later to underline the other important things which are also there. So, we have the dry outer layer, the process, what they're trying to talk about here. So the dry outer layer is removed from the onions, and then you have a sodium chloride solution which is added for a few hours. And then it says that you remove the sodium chloride solution and you put it in a solution of ethanoic acid. Right now, my suggestion, as I clearly pointed out, please do not think too much here about, should I not? Just try and work on this part. So as we move ahead, it says it shows a container of pickled onions and some onions which are fresh up. And now we move on to the questions part which are there. Let's go down to the question part. So the first question which comes in is, what is the independent variable in this investigation? Now, how do I answer this? The answer for this question, the question for independent dependent variable, is right above in the question which is given to you right here, and it is right here. So some students decide to investigate how placing onions in different concentration of sodium chloride solution affects the mass of onions. So here is your statement or the hypothesis of the experiment. So it's asking different concentration of sodium chloride affecting mass of onions. Different concentration is the independent variable, mass of onions dependent variable. So the answer here is going to be concentration of sodium chloride solution. As I mentioned earlier, it's always mentioned in the question, so it's very easy to score in this part. Okay, let's try and move to the next part. Okay, as we move on to the next part of the question, the next part is the table question. So they've already made the things bold for you. about what they think is clearly very important. Okay. So here they have, as you can see, if you remember from your AS concept, this is a concept of proportional dilution. And it says students are given a stock of about 20% stock solution of sodium chloride, and the student decides to use 0%, 1%, 5%, 10, 15, and 20%. Students make 140 centimeter cube. So 140 is your total solution which is going to be made. Complete the table to show how 140 could be made by using proportional dilution. As I mentioned, this is an example of proportional dilution. So we are starting from 20% stock solution. Okay. And how much of this 20% should I take so that I can get 140 ml or centimeter cube of each one of them? So what value do I have to change here? It is this value which will I be changing. That is the 15 or the 10 or the 5, and I'll be getting value for each one of them. Now, what are the values which I'm trying to say here? So how will I calculate x? X is equal to 140 divided by 20, which is going to be fixed for all of them, which is a factor of 7 here, right? And I have to multiply it by 7. So if you look at my table, how much should I take if I'm trying to make a 15%? What is 15 multiplied by 7? Okay, that is what your answer is going to be, which is equal to 105. Okay. So please make sure you write 105.0 right here. This is something I had clearly pointed out. This is something which Cambridge is very particular about. Please see, they have used 2 significant figures or 3 significant figures, and after the decimal place, they have a point 0. So point 0 has to be there. Now, when I do 7 multiplied by 10, I get a 70.0 here. If I do a 7 multiplied by 5, this is a 35.0. Then this is a 7.0. And obviously 7 multiplied by 0 is 0. Now, to make a solution, what do I need? Apart from the sodium chloride stock solution, I dilute it with only one thing, and that is water. Okay. Volume of water, which is going to be there. Okay. So now, this is what we usually do. That's what comes to our mind. But when Cambridge is asking you to answer the questions here, there is something important you need to remember. We cannot use any kind of water right here. We need to either write distilled water. So these are a few things which Cambridge is very particular about. And please make sure you write distilled water or you write pure water. Just water will not get you marks. Now, how do I calculate values here? These values are the remaining water which has to be added so that this plus this should be equal to 140. So here, if I'm adding 0, so the amount of water I'm adding is 140 here. If I'm adding 7 here, okay, so again my answer will be 140 minus 7, okay. So 140 minus 7 will be 133.0. Again, as I mentioned, please make sure that you don't forget to do that 0.0 while you are trying to answer those questions. So coming back again, so 140 minus 35 will be 105.0. Okay, and that's why if you got 105, we had 105 in terms of 15. So here we have a 35. Here it's 70.0. Okay. This is how you complete this table. Please, guys, this table question and the one which was just above it are very highly scoring questions because most of the answers are right there. And most of the students don't make mistakes in calculations here. The only mistake they make is not putting the decimals properly or not writing distilled water or pure water. These are very silly mistakes, but these are things, if you keep in mind from day one when you're trying to attempt the paper, it's going to be very, very easy for you to be able to do that. Okay. So let's move on to the next part. Okay. And the last part which was there in this column was the 20% stock solution. So in case of the 20% stock solution, the 20% stock solution, it's very easy to complete that part of the table. Try and complete it on your own before you move ahead in this video, and try and pause the video before I give you the answer here. You'll be knowing. So it's 20%. It is exactly the stock solution which is given to you. So here I will be using 140 and 0 of water. So here also it should be 0.0. Please remember, this is how they have written. So make sure you have the 0.0 written there properly. Okay. So we have the independent variable question, which is done. We have the table question, which is like a good 2 marks, easy to score. Now we come to the next part, which will be the second part of the experiment. Sometime this comes a little bit later after more data questions which are given. Sometime this comes early in the Feb-March 2024 paper. This came in a little bit early. As I said, your first read has to be so your first read actually can be just reading through the whole thing and making notes of the important points which are there. So here again we have the dry outer layer was removed for 30 small onions. 5 of these small onions were there. They were left in sodium chloride. And the mean percentage change for 5 small onions was carried. We consider these are important points. 30 small onions is an important number I see here. And as usual, these are a few things which came to my mind and I've marked them. I move ahead trying to understand what the next question in this entire process is. And as we move ahead, we've seen the graph, and let's just try and move to the question first. So in the question, they ask you, explain why the students calculate the percentage change in mass of onions. So here they are not asking you anything about what they've actually observed in the graph, which is a normal human tendency for us to, try and understand what is happening in the graph and spend about 5-10 minutes here. Rather, move on to the first question. This 1 marker is not asking you the trend of the graph which is there. They are asking you, why am I taking a mean in this process? Okay, why am I taking mean? You can have this question framed in a lot of different ways. It can be change in mass, why mean is being calculated, why are you taking an average? Okay, why are you taking an average? And these are usually 1 markers. The reason is as simple, you're doing all of this for a valid comparison. You are doing all of this. Please remember these words. The reason you are trying to take average or to calculate the change in mass or percentage change in mass, it's always valid comparison which is needed, a valid comparison. You can further extend your answer if you want. Even if you don't want, you can just write that it provides you with a valid comparison. You can go ahead and point out that it allows for valid comparison between onions which are there in different concentrations of sodium chloride. Okay. If you want to write a little bit more, If you're not sure about what valid comparison you are trying to talk about, please don't write it. It can cause you to lose a mark. Rather, just write down it allows for valid comparison. Percentage change, graph, taking a mean. Please make sure you use these words of valid comparison right here. Okay. So there's this one mark, which is clear. So now let's try and move on to the next question. And the next question here talks about one of the students have concluded and they've given you a statement. The statement which they are trying to talk about here is the water potential of onion cells is the same as the water potential of 4.2% sodium chloride solution. With reference to the information provided, including the results, suggests reasons why this conclusion should not be accepted. Your way to answer these questions, where they ask you in paper 5, suggest reason why a thing can be accepted, why a thing cannot be accepted. First thing that should come to your mind, even before you move on to the result, is has statistical analysis been done in this experiment? Did they write anywhere about statistical analysis? Okay, that's your first thing which should come to mind even before you have a look at what the graph is saying. So let's have a look. Have they done any statistical analysis? Have they given any standard errors? Have they given any data about standard deviations? We don't see anything. So obviously, your first point in this question becomes that there is no statistical analysis. Okay, there is no statistical analysis which has been done in this experiment. Okay, there is no statistical analysis that has been done in the experiment. No statistical analysis done in the experiment. Okay. And please remember, do not start these questions by saying that, you know, this cannot be done because. So they've given you a statement already. They are saying that it should not be accepted. So this is where you start your answer with. As I said, the first point is that there is no statistical analysis. They are saying that a particular result cannot be accepted from an experiment. First question, statistical analysis done or not. Okay. Second thing which comes to our mind is why can a particular result not be accepted? Okay. We will have to think of an experimental error. An experimental result cannot be accepted because there might be some experimental error. Please do not write here that there will be some experimental error. We are talking about A levels here. Cambridge expects you to be precise. So, what experimental error are we talking about? What experimental error can we talk about? So, experimental error, which you talk about here, has to be something very relevant. So, you can talk about mixing of solutions, which could have occurred. Okay. Mixing of solutions during the experiment might have occurred. Okay. Secondly, or measurements were not done accurately by the individual. That's an experimental error. So, measurements not done accurately by a particular individual, that's one of the things. Please make sure these two are very fixed answers for a particular experimental conclusion to not be accepted. First thing, experimental error. Why is it occurring? Because of solution, some measurement problem. You can go into the details of each of the measurement problems which were done. Here they've talked about different time intervals, 2 hours, 48 hours. You can talk about how time might not have been measured accurately. Now, what else that can happen on a graph that we will not accept it? Are there any anomalous result on the graph? Okay. Do you see any anomalous result? With my answer, I'm moving to the graph and let's have a look at a third point. Can we see at any anomalous result on the graph here? Yes, there is an anomalous result at 48 hours, right? We see that at 48 hours, there is an anomalous result where there's a percentage decrease in mass, then there is a certain percentage increase in the mass, right? So point out any one of the result which you think does not correspond to the one happening in 2 hours. So if you see at 2 hours line, the one which is a 2 hours line comes down, goes a little bit up, but here you can see that it's going up and then it's stabilizing. So let's say maybe at 2 hours it's going up and down, and that could have been an anomalous results. So let's pick up our third point right here. Okay, let's pick up our third point right here. So let's refer anomalous result was found at about if you go back to the graph, have a look at it at about concentrations between 5 to 15. You can see how it's going up and down for the 2-hour value. So make sure you write that about 5 to 15 percentage change of mass for the 2-hour study. Okay. So we are saying that one of our experimental result is kind of not accepted. Okay. And the reason we've mentioned is very simple. That is, there is no statistical analysis done. There's experimental error. There can be anomalous result. Now we are still left with one last point for us to be able to get a particular answer. And that is something which you will have to make up at this point of time. And you will have to think of the conclusion from the experiment. And this will vary from each experiment. This is something which is not fixed like the other points. As we said that we cannot accept a particular conclusion. So here we can say that the conclusion which they have given, okay, this conclusion which they have given, okay, is only supported for onions, okay, is supported for onions only when the study is done for long hours. Okay, you can see that the 48 hours gives you that reference of result, yes, and is able to be accepted, but most of these conclusions are only supported, okay, when it is left for long hours. If it's not left for long hours, you can see how much percentage mass it's actually decreasing on. So if you have a look here for a 48-hour study, okay, we can say that yes, okay, that 4.2%. So 4.2% might be the concentration that we are talking about. Okay, that we might be talking about, but obviously it can only be concluded when we keep it for long hours. What about smaller hours? So this study is only with respect to a lot of hours, but not for a few minutes. Okay, so this is your 4 marker. I know this is difficult to write when I'm first explaining it to you, but please try and understand when they are asking you that a particular hypothesis cannot be accepted, you do not have to go back to the graph and start explaining the graph. Rather, you have to try and understand here that they are trying to ask you why a particular experimental result will not be accepted, even though you can see that it is happening. what can go wrong in a particular experiment. Think of that as in that way. So here we talk about very general points in the first three pointers. That is, there is no statistical analysis, there is experimental error, there is anomalous result. It is only the last point when we pick up a pointer from the data which is given to us. That is, the conclusion is only applicable when we do the experiment for hours and not for very less time. Okay. So that is one of the things which we are talking about here. Let's move on to the next question. And when we move on to our next question, most probably our next question now talks about the big 6 marker which we have. From my previous videos, if you remember, for the 6 marker, I had given everyone a small hint. So here they have a turnip, which is a root vegetable. They've put the C part here, and they've said that there was an initial test which was done to block osmosis. And now they ask you to design an experiment. The crux of this paper, the student, please try and read what they're asking here. This is very important here. As I mentioned, you have to underline the point. Okay. They're asking you to describe a method that the students should investigate on the effect of increasing temperature over a range of 10 to 50 on the rate of osmosis. So, independent variable becomes increasing temperature. Dependent variable becomes rate of osmosis. It says do not include any risk assessment or how to calculate the rate of osmosis. Okay, your method should be in logical order. Please remember the order has to be correct in this. So please make sure that this is done in a proper manner, in a proper logical order. Now coming back to how are we going to attempt this question. So when we had the planning question, I had given you that there are always two fixed points which are there for this 6 marker, 7 marker, whatever comes in. One is that of risk assessment, where you have to write the kind of risk it is, what is the risk that you will be taking, and what is the safety precaution that you will be using. But obviously, we lose our point here because they have clearly said that you do not include the risk assessment here. We still have a second savior point, that is repeat. Okay. So out of the 6 points here, the sixth point is already fixed for us, where we say that repeat the experiment with each temperature at least 3 times. Okay. So here it's important that you repeat 3 times and we calculate the mean. Please do not forget to complete the sentence. If you just say that, okay, I'm going to measure it 3 times and I don't write that I'm taking a mean because it's obvious, it's not obvious, please, you have to write this. Because in this question, out of the 6 marks, we already know that we've lost 1 mark, which they've asked us not to include. Now, coming back to our first point, which has to be about the independent variable. Okay, so we use 5 different temperatures. Okay, always using 5 different independent variables. I'm using short forms here. Please do not use these short forms in the exam. Please remember, do not use the short forms. Use 5 different temperature: 10 degree, 20 degree, 30 degree, 40 degree, and 50 degree. So you've mentioned 5 different temperatures. That's your first point, which is fixed. We've already got 2 out of 6. Now, I have to measure the temperature, right? I'm saying I'm going to take 5 different temperature. So how am I going to measure the temperature? Thermometer, okay, is used to measure the temperature. So first 2 points are about, I'm repeating again, first 2 points, how many independent variables and how am I going to measure the independent variable. Now we come to our constant variables. That is, what are we keeping constant in this process? So obviously they said that we are taking turnips, okay? So we make sure that we have the same age of turnips. Okay. So if you say same type, same age, same variety, all of that comes under one. So that's something which we have kept constant. We can pick up an other constant. So they say that they describe a method to investigate the effect of increasing temperature on the turnips. So we cut equal size turnip pieces. Okay. We make sure all the pieces which are cut, okay, they are the equal size turnip pieces. Okay. And the last one here is, how am I going to measure that the rate of osmosis was different, and how do I study that? How do I measure my result? So at each temperature, how am I going to measure my dependent variable? So at T temperature, I first take the initial mass and the final mass. Okay. I measure the initial and the final mass. Then only I will be able to say that, there was loss of water or there was rate of osmosis. Okay, so here we are done with the 6 points. It's not that difficult. Here in this question, in the March 2024, it was different because they asked you to remove the risk. And a lot of students did the mistake of writing a fixed point, that is the risk. Please read the question properly. We still had a repeat experiment, which was always there. We start with the independent variable. What independent variable do we take? 5 different values of it, whether it is temperature, concentration, whatever you're taking. How are we going to measure it? That's your second point. The next 2 point can be your constant variables. Okay, I'm repeating again, and please do not forget to write how are you going to finally measure your dependent variable. A common mistake which most of the students do is they write about the independent variable, they write about the constant variable, but they forget to write about how are you going to present your result. They clearly say that it has to be in a logical order. You are saying that we are going to do an experiment this way, but you're not telling them how to measure the results. That will not make sense when we are talking about a logical order. So please make sure 2 points about independent variable, 2 points about constant variable. Limit yourself to that. At least 1 to 2 points about how are you going to measure your dependent variable. And the last 2 points are fixed for the risk and repeat. Please make sure you read the question. Have they asked you not to write one of them as mentioned in this question? Be very, very careful with this. Okay. Please, everyone, if you look at the thresholds from every year, paper 5 is a highly scoring paper. If you're able to plan it out well, practice as much as you are able to do it, it'll be easy for you to attempt this. So let's start with the next question. So the next question here is the graph question. In the March 2024, this graph question as well as the planning question has come quite late as compared to our earlier or previous past papers, but this question is going to be there. The planning question, which is a 6 or a 7 marker, is also going to be there. It can be in the front, it can be towards the back, but they are going to be there. And these are one of the scoring questions where you know where they are very easy to attempt them. So please try and work around these questions, practice them as much as you can. Coming back, this is a two marker, as I had clearly told you. When we are talking about a two marker, 1 mark in my previous videos, as I mentioned, is about labeling the axis. So repeating again, we have effect of increasing temperature. This is our independent variable. Rate of osmosis is dependent variable. Okay. So as ABCD goes, dependent D comes before I. So dependent comes before independent. So we have effect. So we will have effect of increasing temperature. So please do not write effect of increasing temperature here. So temperature slash degrees Celsius is what we want to be written here. In temperature, mind, it should be as a full form, not the short form, which I have written right here. Here is your independent variable. And here we come back to our dependent variable. Okay. Our dependent variable is rate of osmosis. Okay. Rate of osmosis. Please be very, very careful whenever you are drawing this graph. So your rate of osmosis is usually never zero. Okay, so we are looking at temperature from 10 degrees. So we are looking at some reading here at 10 degrees. Also, we will have diffusion. So as the temperature increases, the rate of osmosis increases. This is how your graph is supposed to look like. A very simple graph, not too much work. As the temperature is increasing here, so is the rate of osmosis. No dent, as you can see here. Please make sure it's a straight line. Do not do this mistake. And very easy to score 2 marks. A very fixed question. You should remember which side are the axes, independent, dependent. Please make sure before you are going for paper 5, you have that clearly done with you. Okay. And now we move on to the next one. Okay. Now we move on to the next question. And the last question of question number 1, it says suggest one hazard. Okay, one hazard. So here we are coming to the error type questions which used to be in the AS level papers. Also here they are asking you the error question, the risk question. So it should be easy to associate and try and find an answer here. The answer we are looking at is what hazard can it be? Okay. And what risk can it cause, or what can it lead to, and what is the precaution you should be taking? So while you were working with the effect of temperature on the turnip, we mentioned that we'll be cutting the blocks for turnip. So here, very easy, the sharp object which is right here. Okay. We have the knife, okay, or the blade, whichever one which you will be using. It can cause an injury, and obviously how you are going to cause, how are you going to try and avoid all of this? You can use a board to cut, use a board or a tile. Okay, while you are doing the cutting, you can use a board or a tile. Okay, now usually we try and look at things. Okay, a very common answer which everybody wants for precaution is to wear gloves. Okay. So now if I want to wear gloves, what kind of injury am I looking at? Risk am I looking at? It can cause an allergy. That is why I'll be wearing gloves. Now, what can cause an allergy? I have temperature, which I'm looking at, okay, which is the hot water bath, which is there. So obviously the hot water can be there, or I have turnips, which are there. So these can actually lead to So the more obvious one which comes to your mind is usually this part, which is also correct. And if you can come up with this, so you have to only come up. And please remember, if each one of these is correct, then only you will be able to get that 1 mark. Not that difficult to do. Please try and answer it. If you can't think of the hazard directly, think of a precaution first, which you already know, and you'll be able to come to the answer you're actually looking at. Okay, with this, I will end my video and I will continue with question number 2. Okay, I'll continue with question number 2 from the Feb March 2024 paper. Please remember the question number 1. Again, I'm repeating myself, is a very fixed one, talking about independent or dependent variable, asking you about a graph, asking you about a planning question. Okay, please make sure you have information and you've practiced all of them, how to write these kind of questions, as I will keep on doing more and more paper. Do not forget to like my videos, and I promise I'll be uploading more. Take care. Bye bye.